My name is Vanessa Lorenzo. I'm based in Lausanne, Switzerland. I'm originally from Spain, and I made. I'm educated as a design engineer, industrial design engineer, and also as a media designer and interaction designer from Geneva, from the School of Arts in Geneva. And I am also part since 2014 of a community in where I live in Lausanne called Aquarium. Aquarium is an open public laboratory for doing science-related projects and biology in an open collaboratively way. We call it DIY biology and open hardware as well, open electronics, using all kinds of open source gear as Arduino, Raspberry Pi, and also all kinds of upcycled materials. And so at the end, the objective of so here is where I started developing a great part of my work because I had access to biology. And since I was coming from the digital media design part from the school, I started mixing both worlds of using electronic media, sound, video, storytelling on one side to try to communicate with the more than human. And I don't know if you saw my introductory video before, where I describe the more than human as something that is not human. So it can be insects, lizards, animals, trees, plants, algae, microbes, fungi. It can be anything. And I guess that at the end, my question as an artist and researcher, hybrid researcher, is to wonder how we could change the way we perceive the more than human other in a context of ecological crisis through biomedia practices. And if this approach of going with this electronic or digital media to interact or try to communicate or try to speculatively listen to the more than human, if there is a change in our behavior, in our perception, and therefore some kind of also political change towards the way we relate to the more than human other. Since this meeting has a lot of international like public and especially from India, I must say that I'm conscious that I'm talking from a Western perspective, so from an European perspective of how we relate to nature and to the environment. And I'm aware that, or what I try to do through this work is to try to change this human exceptionalism perspective, which is the thought that humans are in the center of the cosmos and they are somehow above every other being, and we can fix every problem with technology. So it's a sort of critique to how we ourselves see the more than human other and nature, and to be conscious that we are part of an ecology. And yeah, at the end, it's also about deconstructing these kind of identities and looking us ourselves beyond our preconceived subjectivities, categories of species, human species, genre, and every other category that you could imagine. So we could allow other stories and dialogues between nature and humans. Maybe you can introduce yourself, Ajit.
Yes, I'm Adit. I'm 7 years old. I am I I am homeschooled, which means that I don't go to a school. I don't go to a school. I study at home, mostly by myself. I love to read and study animals and also make things. I also save and study spiders. Now, I have a quite interesting example of a tropical spider in this box. Let's see if I can show her to you. Come on, you little girl.
That's awesome.
It's a Telemonia dimedactyl. It's a Telemonia dimedactyl, and a quiet large female. A quiet large female Telemonia.
Wow, so beautiful.
I play music to her. She's a jumping spider.
Wow. That's the most wonderful thing I've seen in a while. Oh my gosh, that's so cool.
I also
how do you
blogs about nature
mm-hmm.
and stuff like that, and mostly poems. But sometimes I write stories, and one of my recent stories was about a jumping spider.
I don't know much about jumping spiders. Where have you found her?
My father, it's the
Where did you find the spider?
the spider. I found her sitting on one of my mother's clothes.
I must confess that I am afraid of spiders. I was afraid of spiders, very afraid. And now I'm starting to like them a lot. And I don't know why. I was always thinking like spiders are like human hands. And that's why I really I was afraid since I was a kid. And now I'm not a kid anymore. I'm 35. But I'm still learning to relate to spiders. And actually here at my parents' house, there is a little hole in the balcony, and they feed the spider every day, and they call it Mrs. Tekla. And it's 2 years now that they feed her every day. And now there is no more Mrs. Tekla, but there is a little one. And so they believe that is her daughter. So I find these stories very, very interesting, because when we see a building like ours, a concrete building, we never think about the non-human inhabitants that are living in them. And actually there are a lot of mini tiny apartments inside a building. And I always laugh about this story. Do you have also this kind of more neighbors apart from the spider?
Well, not as such, but we have lots of tropical butterflies, and well, it's quite a national place. In India, before the coronavirus, it was like quiet, like polluted and everything, which I didn't like since I wanted to see the stars outside. But now I can see the stars because there's hardly any vehicles on the roads right now. So there are more uh, like jumping spiders and butterflies and other sorts of things. I even found dragonflies. This one was dead when I found it, so I've just preserved it so that I can study it. Uh,
Do you
this,
have a microscope?
this is yes, I do have a microscope. Uh, I also preserve some of my pet spiders.
Okay. And what was that? I couldn't see it on the screen.
Oh, that was a preserved dragonfly.
Ah, yeah, I see that. Wow. And what is that? A resin?
Yeah, that's artificial resin. I've even preserved the pet spiders, two pet spiders of mine. So this is one of them. Okay, so this black little spider that you see on the right, he was my old Hazarius dancer knight before he died. Poor old little Hazarius.
I was, wow, I was muted, sorry. That's awesome. I have a little question here in the chat from the inquirers. And they ask, what's the scenario with the technologies democratizing scientific biotools, like, for example, microscopes and so on, for anyone who wants to use it, especially artists? Okay, so remember when I was asking you if you had a microscope?
Yes.
So that's why I asked before, because normally not many people have access to some kind of tools that are normally available in laboratories, and so one of the reasons why I joined this community in Lausanne is because I didn't have a lot of tools to do scientific projects, and I learned that through this culture of DIY biology or DIY science and hacking, we could make some tools like webcam microscopes, laser microscopes, and shakers to mix reagents or chemicals, and incubators with some heaters, so we could culture fungi or bacteria or keep algae cultures at a certain temperature. And so I guess this question is about what is it like to have access to all these very nice machines, to see nature at the end, no, or biological material as well. And for artists, I think, like me, it's something very powerful, because you know, you not only can learn how to culture or to take care of insects or bacteria or fungi, but also I found another palette of tools to work with, which is the scientific tools, and also biological matter, and insects and bacteria, etc. So for artists, I think, or I would say, at least for me, it's a world that opens up to express ourselves. And yeah, for example, I could paint with bacteria, you know, like there is different colorful bacteria that we can grow from our skins, yellow, orange, pink. And there is also some bacteria from the skins of other animals and insects with have wonderful colors like violet, green, red, etc. And when I access to this community and to these tools, I was able to, for example, print with bacteria, make designs and print them on layers of gelatin with bacteria. And so I guess and all this knowledge is open source, so it could be spread to other people, artists or not artists, and also scientists who were curious about it. So I guess having access give a lot of empowerment to the people who use them, and also as an artist it gives a lot of possibilities to tell stories with moss or with other kind of natural beings.
Speaking about your project where you printed designs in gelatin with bacteria, that's kind of what Robert Koch did. He like took these needles, then dipped them in bacteria, jars brimming with bacteria, then like chased them on agar. Then the bacteria ate into the agar, and then they formed the designs which he made.
Mm-hmm. Yes, this is one way to do one way to do it. So I don't know what you used, but I used silicon plastic, and we could make a stencil cutting the design with a laser cut, or you can do it also by hand with a cutter. But also after a year, I had a grant to work on a project to upcycle a 3D printer, which I don't know if you know these plastic 3D printers. And okay, and there was one that was not working at all. And it has the same stepper motor as a peristaltic pump. So a tube that was sucking liquid from one side and put it into the other. And through a needle, very, very fine Pasteur pipette, like a glass needle, and hacking the code of the 3D printer. You can design a logo or write in a very simple manner with points on the screen, and then the machine can make the puncture with the bacteria on its needle, and then in a very DIY way you can take the bacteria as a post-representational tool, something you can turn bits of digital media into bacteria, into molecules, into something organic, right? And okay, I see that there is another question from the inquiry, Dia, and it says, have you ever tried any experiments on your own biology? I guess this is referring to, and correct me if I'm wrong, if I've tried to do experiments with my own body. Yes, I've tried. I took, as I said, bacteria from my skin, and I cultured different colors. And with these colors, I made different mixture with bioplastic and different printing techniques, and and also different culturing methods with different culture media. And right now, I want to go a little bit farther, and I'm working with skin culture in a project called Mari Mutare, for which I received funding from Helvetia, and also I'm doing a residency at Angar, Barcelona, and also I did a residency recently at Utopiana in Geneva, and it's about ornaments based on the figure of a hybrid, and a hybrid between a human and a plant. And right now, I'm learning how to make skin cell culture on a petri dish, and very soon I will probably learn how to make a biopsy and try to culture my own cells in the lab. So I don't know if you can imagine the situation, but imagine that you can take a piece of your skin and make a large patch of your skin outside. And that is something that it raises a lot of questions and a lot of curiosity, and also questions about ourselves, where our body ends and where another entity starts. And so there is a lot of questions about identity and who we are through doing this simple, but through this question, right? And trying to experiment with biology in that sense.
Now you want you make me want to grow my own spider cells.
That's also a very quick. It's a very good question, because so at the end, the context of doing DIY biology in an open laboratory, first of all, is to be open and to share and to discuss about all these particular questions, and also the ethics of it, right? The ethics of creating our own little universe with our own species. And I think it's very healthy to have a conversation about what if this could happen, and what would be the consequences of doing all these experiments, right? And I think it's very interesting, and it's very constructive to have access to start this kind of experiments. It's always very difficult to achieve a result, and for me personally, is not the goal to have a real living human plant hybrid walking around in a shopping mall or in the streets. But I like to provoke these questions of what if, and if it's my body, what would I, how would I recognize myself if I change myself? If I make my own spider, a new spider, would it be still the same spider? And what would be the consequences if I release that spider into the wild, and so on and so forth. So normally these questions are very are happening in a very hermetic environment in laboratories, and when we open this to the public and to the people like us, and having the tools to realize these experiments, we have actually the opportunity to learn more about it, and to inform ourselves about it, and to have something to say about it. I don't have any more questions here. I don't know if you want to tell me your next projects, Adrit. What do you have in mind?
Well, my projects are that I want to start growing rhino beetles. Rhino beetles, like those big giant horned beetles uh, with this tusk that they used to fight other males for females. Well, I'm thinking about growing those and also stick insects because they're very popular. So,
Wow, sounds amazing. I love these huge beetles with these horns. I think they are quite also strong, right?
yes, quite strong. A Hercules beetle, which is a rhino beetle, it's about like around 1 foot in length. That giant. It can lift up to 850 times its weight, which I wish humans had. If a human had it, then they could lift uh, giant like buses and buildings just just like that. They could just lift buildings without any without a lot of effort.
That right now I had an idea, and I made an experiment with people from Shenzhen last year, with students from the University of Tsinghua, and it was very nice. The outcome, and the experiment started with the question of if you had the chance to have a capability or a power or a characteristic of another species, it can be a plant, an insect, an animal, what would it be? So if you had the chance to hybridize yourself to become something else, some other, more like some other species or animal, what would it be?
Myself, well, I'd rather it be a clouded leopard because that's my favorite. That's one of my most favorite mammals. If the clouded leopard was extinguished by then, no, not even a trace of the genes, then I'd rather a rhino beetle or a spider because, like spiders, they never got extinct. They survived the dinosaur age and the meteorite crash and whatever happened, but it killed those giant creatures away, the dinosaurs, when they were so mighty and like conquered the whole earth. And
Wow.
they survived along with most of the other invertebrates. And then the mammals, the spiders wasn't even prepared, but they still made through. Now the human age, still many spiders have managed to escape uh, death in forests and stuff like that. Even if the forests are extinguished, they survive. They're kind of very smart.
That's very clever, actually, because you're sure that you won't be extinct. That's very clever. I would like to become a hybrid with a vegetal or a plant. You know, I've been using these technologies, you know, these filters, digital filters, to try to hybridize myself and to try to imagine myself as a plant. And I really actually like it a lot. And actually, you know why? Because, you know, a plant uh, needs the sun to get nourished and to make the photosynthesis. And imagine that we could get nourished from our skin, just sunbathing. so be we wouldn't have to go shopping, right? Or shopping that much, maybe. And I don't know, maybe imagine that we become fungi, and we can uh, allow the dialogue underground for all the forest, and help them, the other plants, have their minerals and nutrients. But at the same time, we could keep all their secrets and gossips because we know what they're talking about. And so we become a super powerful agent of the forest. And yeah, I don't know. I think I would like to be, for example, like the moss that I have behind me.
Yeah.
I would really like to become a little bit more like a moss because they live super long time, and they are witness of all the changes in their environment because they live so long, and they are highly efficient. They adapt to any conditions. They can adapt to harsh conditions, to very sweet, cozy, warm, humid conditions. um, and they were actually the first plant to jump out of the ocean to start growing on earth. So I think it's like our kind of ancestor in a way. So I would like to become a little bit more like a mouse.
Uh, I kind of like that sort of idea. Then, uh, if we just look like normal humans, but we had photosynthesizing technology in our skin, then it wouldn't like we even poor people would get some nourishment at least every day if there wasn't if there was sunlight, because uh, plants need the sun to like make their food. So there will be literally no poverty. Then money would have no use because people use money to buy foods and things that they like and stuff like that. But in those sorts of worlds where people just eat stuff, the money would have literally no value.
Yes, and that would change the whole system a lot, and the whole value. And maybe knowledge would be a little bit more important than anything else, and values as well. I have another question from the chat and from the Q&A. For the Q&A, I don't know if I have to answer them now, um, but there is a question that says, can you share a project that scares you in the sphere of DIY biology? So I guess this means if I know about any project that is scary in this area. I think it's a very interesting question, and I think it's linked to our conversation before of what if we could make our own species of spiders, or our own species of hybrids, animal, people, something, whatever. Um, I think what scares me of not only DIY biology, but biology in general, is to not be transparent about it, and not to do it secretly for not good reasons. I think either DIY biology and biology, that has to be transparent, and even if it's activist, or it has a political discourse. Like me, I'm talking about like an artist, as an artist, that I often have ecology or feminist or ecofeminist approach. um, I think it's for, at the end, it's for a common good. And what scares me is that biology or DIY biology is done in a hermetic space, in the very private laboratories, very powerful, where there is no transparency and not control from neither the citizens or uh, the community around. That's my answer. But there is no, right now there is no DIY biology project that really scares me. I think what scares me is what can we do with massive amount of data that we can take from mass genetic experiments, like for example the PCRs that we are doing right now because of the pandemics, or all the data that you can collect from the people without telling them what is the purpose behind it. And of course, yeah, the topic of designing babies without control and all this kind of stuff is something that scares me a little bit. But I'm confident that if we are all transparent enough and we have the tools to regulate, but also to trust that the people that want to learn, they are not thinking about doing harm by default, but to really learn and to foster curiosity. And I think an educated community in society is always a better community. I think we have to move to the collective.
I have
Yeah.
Hi, Vanessa. Not yet. Okay, my
Okay.
internet connection has stabilized finally.
Okay.
I have a question. Do you have any recommendations for the experiments that I can do at home, and where do we procure the materials and chemicals for those things?
Yes, that's a very good question. I know that you can go to a webpage from Aquarium and Hacteria. And I think there is also a community of DIY biology in Bangalore, if I'm not mistaken, which also makes art and science projects. I don't know. Sayed?
Shaira.
Shaida?
Yes.
Yeah, sorry. Can I have the contact of or maybe of Adrit, or maybe
Sure,
he
of course.
online? I can send you a lot of information of where you can go to build your own basic laboratory with the basic materials to start projects. And I see that you're quite advanced already, and I'm very happy and proud of you, because with 7 years it's very impressive. And I hope this information is gonna help you a lot. And I'm gonna see your results as well very soon.
Please do send me the
Yes.
information. It would be wonderful if I had stuff like that. And well, about Mars, you're making me think about growing very own very own culture of Mars in, like, if I was to grow Mars in a but in something like this, would Mars manage if it was put into something like this?
Yes.
It's because it's quite a tiny bottle. I usually
Mars.
uh, yes, I've also done some sort of a experiment. I'm trying to like grow a embryo outside the seed. So now what I've done is, like, for example, if you open a embryo in the pea, then on the sides of the embryo is the coating that makes the food for the embryo. So I've thought about that. So I've like mashed up these peas, and then like I've put them in the and then I put the embryo inside directly after it was opened from the piece. So I've done it. It's still not growing. So
Wow. I'm sending you the links that I told you about before through the chat.
Adrit, after this, we're going to move on to a Q&A. So I'm going to, if you'd like to stay in the room, you're welcome to. But we're going to close in about 10 minutes, and then Vanessa is going to move to the panel discussion.
all right.
All right.
All right.
So Vanessa, I've put down a few questions, another 2 questions as well in the chat. If you could maybe take a bit and.
Okay. I read now. If I can share any sources that inspired the project. I'm sharing Hackeria and Hackarium wiki pages, where you can find the projects right now. So it's over there. But anyone, it's very welcome to contact me through the links that are on the webpage of the Critical Conditions Domain of Nature. And for anyone that maybe it's watching this video live, and also my own webpage, where I also have some links to projects with moss and printing with colors and bacteria, and some workshops that I made through the last years with different communities about the quality of soils and water and microscopes, yeah, etc. But I think like the most important thing is to find your nearest community that is doing some hacking, even if it's only well, only even if it's electronic hacking, it's already fine, because the mindset of doing things for ourselves and sharing and building little machines to or little experiments is already there. The seed is there, and I think actually in Bangalore it is already a nice community, but I need to check. I think if you go to Hackeria, you will see the links that they already made with an art school there. What else? I don't know if we have a little bit more time.
We have about 5 minutes. Yes.
Okay. So there is another question that's, do you think DIY biology will become commonplace in the near future as a consumer product to make us more than human? I think that we are already more than human, because we have a lot of bacteria and fungi and other stuff inside of us. But yeah, I think at some point there is some people. It's not what I practice personally, but I know that in DIY biology there are some communities that are pushing the limits for self-modifications and glowing stuff and so on. Mm-hmm. It's not my interest, but I think it may happen. It may happen.
I have a question.
Sure, go ahead.
It's a pretty short question. It's related to man-made stuff joining together with natural stuff. I've heard a lot about the scapulary effect. Like my mother showed me the stuff on filter paper, like chromatography. So I've done those experiments with her, and I was wondering if we could if we took a flower, because the flower sucks up liquid if it's placed in a vase. So I'm wondering if I attached a water soluble thread, something that got wet when it was exposed to water. If I put a thread up the stem of the flower, then left it in water, would the flower survive?
I didn't quite understand. What happens with the water up the?
Okay, so now I'm just going to draw something.
No, you just
So it's like if this is a flower and this is the stem, just imagine a stem over here. And then if I took this very thin thread that got wet when it was exposed to water, then I fitted it up the stem. If I fitted it up the stem and then placed and then gave it some contact to water, would the flower suck the water up from the thread and would it stay alive?
I don't have experience with that, but my answer is always try it out. Always try it out. And if you fail, try it again and and fail better. That's what I would say. Sorry, I cannot help you a little bit more, but this is how also I learned about biology, and asking the people around and trial and error, and not being frustrated because there is not the expected result, but being excited about trying again and getting the information from the people that knows. And I think that's also linked about the previous question about the near future experimentation. I think as long as we're eager to learn and we are respecting our ethical chart and being transparent about it, and we can do any kind of sexy, crazy, incredible dream projects.